Bioinform takes Particles 3. This session explains data retrievals in biology. Biological data flooding in the last decade. So researchers are finding it very difficult to find out the required scientific data with ease. Simultaneous development in bioinformatics produce many softwares and tools to retrieve biological data with ease and pave the way to develop biological and related science fast. This graph shows how the biological data flooding for the last decade, 2017 to 2018. In 2007 the nucleotide data was only 10 raised to 10. But in 2018 it reached 10 raised to 16. This flooding of biological data require a better data retrieval tools and servers that we will decide in ahead. So watch the video completely.
Let's see various tools for web search. Google Web Search. Google is one of the most commonly used web search tools, both research purpose and general purpose. It is free and unlimited for retrieval of any kind of data. As an example, the World Human Genome Project retrieved 16 crore and 10 lakh results within 0.89 seconds. This figure shows the relation between entrez and other resources to retrieve biological data for various purposes. The explanation for various resources are given are hard.
This table shows description of various entrez database. PubMed for biomedical literature. Nucleotide sequence database. GenBank protein sequence database. 3D for macromolecular structure. Genome for complete genome assemblies. PubSat for population study set. OMIM for online Mendelian inheritance in man. Taxonomy organisms in GenBank. Book for online books. 3D domain. OMIM from interest structure. Unis TS marker and mapping data. SNP for single nucleotide sequence data. CDD for conserved domain. Journal for journals in entrez. UniGene gene-oriented clusters of transcript sequences. PMC for full-text digital archives of life science journal literature. NCBI website for NCBI web search.
This the PubMed search for the word counts and metastin. It retrieve many articles. You can select all or any one interested. Then click on the article you want. It will lead to the entire article. This is one of the complete article retrieved from PubMed. You can download the article or read it or copy the citation separately. Sometimes they request for payment, but most of the articles are free of cost. Another very important benefit is you can retrieve many reference article from this one article.
Data retrieval and submission tools on the NCBI. 1. Entrez provide integrated access to nucleotide and protein sequence data from over 100,000 organisms, along with 3D protein structures, genomic mapping information, and PubMed Medline. 2. Linkout, a registry service to create links from specific articles, journals, or biological data in interest to resources on external websites. 3. Cubby allows entrez users to store and update searches and to customize their linkout display to include or exclude links to providers. 4. Citation Manager allows to find out the PubMed ID or the Medline UID of any article in the PubMed database, given its bibliographic information.
Another data processing method in bioinformatics is sequence similarity searching. 1. BLAST homepage. Access to BLAST basic local alignment tool programs, overviewed, help documentation and FAQ. The BLAST email server has discontinued. 2. Blink. Display the results of BLAST searches that have been done for every protein sequences in the entrez protein database. 3. Network client BLAST, a BLAST client BLAST 3 that access the NCBI BLAST search engine. 4. Standalone BLAST, download BLAST executable for local use. Let us now look on the taxonomy, related tools and servers. 1. Taxonomy browser. The tool for searching NCBI's taxonomy database. 2. Taxonomy BLAST. Groups BLAST hits by source organism according to their classification in NCBI's taxonomy database. 3. Tax table. Summarize BLAST taxonomy data and displays the relationship of the organism to other through a color-coded graph. 4. Prot table. Provide summary of protein coding region in a genome. 5. Tax plot. Provide a three-way view of genome similarities. Biology Workbench. It is an integrated server both on linear and standalone. Workbench will help the researcher to extract most of the data required to make bioinformatics in silico research success in a big way. It includes protein parameters, taxonomy, gene analyses, protein analysis, gene prediction, DNA and RNA analysis, and many more. As a bonus, I can introduce Biology Workbench. It is very useful because it is an integrated tool for most of the bioinformatics data retrieval. It can be used for taxonomic study and construct taxonomic tree, and for protein parameters study, gene finding, and many more. Download the Workbench and follow the instruction manual strictly. Thanks for watching the video. For further inspiration, I request all of you please subscribe and like the channel.